Good afternoon, everyone. My name's Brianne Rath. I'm the Public Programs Coordinator here at the Nantucket Historical Association. I want to thank you all for coming out to Food for Thought today. Before we get things started, I'd like to ask everyone to please take a moment to make sure your cell phones are on silent so we do not disrupt today's talk. Today's Food for Thought is brought to you by the MS Worthington Foundation, and media sponsorship is generously provided by Novation Media. Today's lecture features the Linda Loring Nature Foundation's Director of Research and Education, Dr. Sarah Bois. Sarah received her PhD from the University of Connecticut, where she focused on invasive plant distributions in New England. As the program biologist for the Invasive Plant Atlas of New England, Sarah worked with and trained volunteers throughout the Northeast to identify and document non-native species occurrences. Sarah's position at the Linda Loring Nature Foundation marries her interests in research and education, where she encourages people of all ages to participate in science and research at their own comfort level. Please join me in giving a warm welcome to Sarah.
Okay. Thank you. Can you guys hear me okay? Okay, that's the first test of the mic, so. All right, well, thank you guys so much, and I want to thank the NHA for having me come out here and I get the chance to talk about some of the work we're doing at the Linda Loring Nature Foundation. And so I'm going to talk today a little bit about research that we're doing at the foundation. It's ongoing, so we have some results, but it's kind of things that are happening, things people can be involved in. And so, yeah, let's get started. So before we talk about exactly what we're doing for research, I like to kind of give a background of why we're doing this research and the type of research we're doing. So we're going to talk about looking for signs of natural events. So when we think of, I know we're in fall right now and leaves are falling, but let's put our minds to spring for a minute. So signs of spring on Nantucket. I always like the osprey. I think that the first osprey coming back in March is one kind of sure sign that spring is arriving. We think a lot about daffodils on Nantucket, so even if you're not maybe a naturalist or someone who observes nature as much, maybe you're not a birder, but everyone on Nantucket is interested in daffodils, the timing of the daffodil festival, are they out, are they not, will they be seen along Pulpus Road or Milestone Road? And we always talk about whether they're early or late. So those are kind of signs of spring and noticing these kind of events. I love forsythia, so that upper right photo. And then sounds, spring peepers. Who's the first one to hear the spring peepers in the spring? And then this poor little crocus. So crocuses are one of those first flowers in the spring. And oftentimes we end up with pictures like this where they're covered in snow. So that's one trade-off of being an early bloomer or an early leaf out plant, as you might end up with some snow on your leaves. Now, all of these plants and animals have evolved to be signs of spring, to be early happenings. And so the study of these types of events, like when the first osprey lands, when we first hear spring peepers, and when the first crocus flowers bloom, is called phenology. And I'm going to use this word over and over again. And it's really just making observations through the seasons. So this example of series of photos is a red maple tree. And so when we look at phenology, we're looking at the timing of something very specific over time. So like spring, like this photo would be like spring bud burst, when those green leaves first bud, when it's a full flush of leaves on the tree, and then throughout the season as the leaves start to change, and then fall and the leaves fall off. So those are all events you can kind of look at. We look at them over time and also based on temperature. A lot of phenology or phenological events are based on temperature, and we'll talk about that a little bit more. But we can also look at the temperature associated with all these different events. And so one thing that we're starting to notice throughout the whole country is that spring specifically, that I'm going to be talking about today, is happening earlier and earlier. Now this map is just from this year, and it's called the Leaf Anomaly Index. So organizations like the National Phenology Network, they put together all the data that people collect on phenology, and they look at, on big scales, like nationally, what's happening with things like the spring timing, fall timing, and things like that. So what we found this spring, this is just for 1 year, the darker red indicates earlier and earlier. So the darkest red is that spring has been happening 20 days earlier than average. The blues are actually spring happening later than average. So it's really interesting, while maybe in some of the national news we might hear about spring happening earlier, it doesn't happen the same in every area. And once again, this is just 1 year, and 1 year could just be an anomaly. It doesn't mean a trend. And I'll point out more locally, a lot of times we talk about New England because we have all this data from big scales. And so New England, if you look this year, was kind of average. For the most part, it looks like, oh, it's about an average spring. When we look specifically at Nantucket, it was a lot earlier, up to 2 weeks earlier, based on the leafing out. So it makes a case for why we should care more locally than looking at just the big regional scale things. So with some of this research, we're looking at interesting trends. So one of the oldest phenology records is from Kyoto, Japan. So people started looking at flowering of cherry blossoms in 800 AD. So it's a pretty good data set, since 800. And since then, we found, I say we, that's a big we, I have nothing to do with this, that cherry blossoms have been happening 10 days earlier. So on average, across all this time, we see that cherry blossoms are blossoming 10 days earlier. But when you look at this line, as it goes down, that means it's happening earlier. The real decrease has only been happening in the last 1 to 200 years. So in our own country, one of the earliest phenology records is cherry blossoms in the DC area. So the National Park Service started monitoring cherry blossoms in 1921. And that's almost close to 100 years. And since then, in that almost 100 years, there's an average of 5 days earlier blooming. So 5 days might not seem like a lot, but that's on an average. So there's some years where there's up to 2 weeks earlier, and some years it might be a little bit later. But an example of the consequences of that is that just this year, in spring of 2017, one variety of cherry blossoms in DC flowered so early, and then there was a late frost, and that whole variety, the blossoms were killed. So that's just 1 year, but if this happens over and over again, you could imagine the consequences for that one cherry tree variety might not be around anymore. And so obviously things like cherry blossoms, where there's so many festivals associated with them, people are very interested in the timing of these things, but it can also have ecological consequences. So when we look more, oh, sorry. I'm going to snooze this. I hope the NHA doesn't mind. When we look more regionally, so this is data from New England, and I just will only highlight a little bit about this. Henry David Thoreau collected, many of you know him for his writings, but he kept extensive notebooks. And one thing he did around walking around Walden Pond, in addition to just his essays and writings, he wrote down the first flowering date for over 150 species that were just around Walden Pond. So I think this is appropriate for bringing up at the NHA that these historical records can still be meaningful data sets that provide us insight into what's happening now and in the future. So what researchers did was look at the plants that Thoreau observed and went back to those same areas at Walden Pond and then looked at flowering dates for multiple years. And this was done in around 2009 to 2011. And what they found, on average, was that for most of the plants, they flowered about 7 days earlier. So this is a New England example of things happening earlier and earlier and earlier. Oh, and one thing I want to add about this is that the lead author on this, Richard Premack, he's going to be here tomorrow to give a free lecture at the Athenaeum. And so I'll have a little slide about that at the very end. So while we can see that spring's happening earlier and earlier, that's great, but what are the consequences? So there's lots of consequences for things like, the timing of cherry festivals, the timing of daffodil festival, but there's ecological consequences as well. Plants can be subject to late frost. In some areas of the country, they might be more prone to wildfires. But one thing I want to talk about is ecological mismatch. So this has become a classic example in science, and I'll run through it really quick. So this is a great tit, this bird. It occurs in Europe. And under an ecological match, there's a suite of species that have evolved to, their timing has evolved to coincide. So the great tit is here, and it lays its eggs based on photoperiod. So birds use the sunlight cues to know what time of year it is, and so they lay their eggs. And at around the same time that the eggs are incubating, there's a hatch of caterpillars. And not just one species of caterpillars, but there's a pretty decent biomass of caterpillars that emerges right at the same time that the eggs are hatching. So this means that right when the chicks are hatched and they need the most food, there's the greatest abundance of caterpillar biomass. That's wonderful. That's a match. Everything's happy. And at the same time, these caterpillars are hatching right when the leaves of the bur oak are young and fresh and really edible. So these oak trees are have a high biodiversity of these caterpillars. So everyone's happy in this upper picture. So in the study, there were observations that temperatures were warming and spring was happening a little bit earlier. And what was happening was that there was a shift. So the caterpillars in general, caterpillars are very responsive to temperature. So as the temperature advances and spring is happening earlier, the caterpillars will hatch out, and there's a high biomass at an earlier time period. The oaks are also responding to the warmer temperatures, and they're leafing out early too. So the caterpillars are happy and the oaks are happy. But the bird is still responding to photoperiod. So when the chicks are hatching, it's still a little bit later, and there's not as much caterpillar diversity available. So there's two things happening as a consequence. They've noted that the great tit populations have decreased significantly because there's just not as much food resources for their young. But then also, the caterpillar biomass has increased, and that has then decreased or further increased the defoliation of a lot of the oaks. So this is an ecological mismatch, and there's just a cascade of consequences that researchers are really just starting to dig into. Okay, so that's great for some of the primary examples globally, nationally, and regionally. But what are we going to see on Nantucket? Are our plants changing their phenology at all, as we know that spring is happening earlier and earlier? Are there any potential species mismatches? Do we, as conservation biologists, do we need to respond? Is there something that we can do? So these are kind of the overarching questions that drive a lot of the research we're doing at Linda Loring. And when I give this talk off-island, I also have to say why it's important that you should care about what happens on a tiny island. And so I often talk about the globally rare habitats we have, a number of rare species. So even if we're not studying necessarily the sparrows and the northern harriers, we're studying the habitats that they live in and are required for their natural history cycle. So our specific research questions are looking at, okay, as temperatures are warming, how is that affecting our Nantucket flora? What is our baseline? So to notice change, we have to have a baseline. And right now we haven't discovered the great historical legacy of phenology data for Nantucket. That'd be great. We have some great bird data for a significant amount of time on Nantucket, but we have to start our own baseline. And then what is our vulnerability of our species? So are there kind of winners and losers to warming temperatures? Are there species that can take advantage of an earlier spring? Or are there species that are more susceptible to early springs and potentially late frosts? So we set up a couple of different experiments I'm going to talk about. One is just field observations. So in order to get a baseline, we want to know what is our quote-unquote normal, what are our normal yearly averages. And so we're looking at 8 different species. We're focusing on common native shrubs, both because shrubs are nice. You can tag them, and they don't move, and you can revisit the same plants every year. But also, if something changes with our common shrubs that a lot of species are reliant on, then we're going to have a bigger cascading effect. It would be nice to look at some of our rare plants, but they're not in as great abundance. So it's it's really important, and I believe it's important to work with common species. Now, in addition to these 8 common shrubs, we're looking at our eastern tent caterpillars. We chose the eastern tent caterpillar for a couple of different reasons. We had some students who were really interested a couple years back in these caterpillars. They go through a regular boom and bust cycle, and they're common native species. A lot of people don't love them, but they are native, and they have evolved to live in these plant communities. So, and when they hatch out and they're feeding on everything, they're looking for these tender leaves, primarily of black cherries out here. We'll see them on a number of different things, but black cherries are their primary host plant. All right, so to orient you to our field observations, just to give you an idea, we're looking at our 8 different plant species, our caterpillar species, and this yellow kind of funny shape is the property of the Linda Loring Nature Foundation. So our property has kind of a funny shape to it. We have about 104 acres to work with. And the green dots are our specific, what we call them phenology plots. So at each of those green dots, we have 3 individuals of each of our study species that are tagged that we monitor regularly. And then also at each of these sites, we have a little temperature logger. So this little thing right here is basically a tiny computer, and every hour it records the temperature. It also records the humidity. And then it's shaded with this little shingle because it's Nantucket and it needs a shingle, but also because it shades it so that it's just reading the air temperature. It's not getting that direct sunlight. And we do that to kind of track what the specific temperature is we can kind of monitor temperature for each phenophase, so each kind of transition for the plant. And here's what we're looking for. So we're monitoring leaves, so bud break, the full leaf out, and then when leaves change color. So this is a picture of a black huckleberry, and we call this like bud break. So these tiny green buds in the spring, when they kind of first start to unfold, that's called bud break, and that's a very significant phenophase to monitor. In flowers, once again, this is a black huckleberry. These are the little flower buds, and right when they first start to open, and then they're receptive and their pollen is available, that's like an open flower. So those are very specific things. And then fruit, we're looking at ripe fruit, whether or not there's fruit drop and that kind of thing. And all these data that we're collecting follow protocols from Nature's Notebook. So this is a protocol set that was designed nationally through the National Phenology Network. Nature's Notebook is their citizen science program. Anyone can do this. It's very easy. Once you do it a few times, you kind of really know what you're looking for. So anyone can do this in their own backyard. They can help us do it. But basically, we're just looking at, and we go out multiple times a week. So at least 2 times a week, but usually 3 times a week in the springtime to look for these changes. And then because we're using the National Phenology Network, we can enter everything online. It makes data entry very easy for our interns and students and things like that. So I mentioned student involvement a few times. We have a lot of, every summer we have interns that collect these data, but in the spring, one thing that's so great about this project is we really focus so much on the spring phenophase, and school is still in session then. So we have high school students from the Nantucket High School. We have a regular program with the Nantucket New School where their 8th graders have independent projects, and they come out and take data and then ask their own research questions. So I just highlight this for this talk so you know kind of how we're involved in the community with this research. And then they have data sheets like this where they have to answer yes or no if they see something, like do you see an active adult, and if so, how many, and that kind of thing. And some of these students were the ones that really got into the caterpillars. So I'm thankful for them for doing that. So I'm going to go through some data slides and then talk about what some of our data might mean for our plant communities on Nantucket. So this is what I would call the beginning of our baseline. So I was saying we're looking for a baseline, like what are the plants doing? And for the most of this talk, I'm going to be talking about the breaking leaf bud. So that's the phenophase we're focusing on, is in the spring when those green little buds just first break open, and that means that they're photosynthesizing and they're starting their season. And so this is called a phenology calendar, And each bar represents when we see that phenophase, so when we see breaking bud. And so we organize this based on what happens first. And so in a normal year, this is one year in particular, but it represents what we see every year generally. That blueberries first, it's a very early leafer. It's got thick leaves, so it's prepared to be an early spring leafer, followed by black cherry, hazelnut, scrub oak, huckleberry, and along the bottom you can see the month of the year. So things start at the end of April, but then they move through May, and then the latest are the beach plums and the bayberries in late May, mid to late May, that are really leafing out. So this is kind of our baseline normal. And we're collecting more and more data every year to kind of bolster this, but this is what we feel good about as our normal. So the thing that we do as scientists is we test the norm, what we consider the normal or the average, like what happens if something changes. And so we set up a twig warming experiment, and we've only run this experiment for 1 year, but we are pretty interested in the results, so we're going to be doing it again, where we're mimicking an early spring. So we can't, we're looking at our field observations year to year and year to year, but in order to see the response of our plants, we want to force an early spring to see what would happen. So with this experiment, we have, for each of our 8 species, we have 10 twigs, and basically we put them in little jars inside at a constant temperature and then wait for leaf out. So that would mimic them like as if it suddenly was warmer and suddenly spring. And so what we're looking for is, this is a black cherry twig, and we're looking for the development of these buds, these green buds, and then the opening of these green buds. And this is a hazelnut right here, so this is leafing out, just to give you an idea of what we're looking for. And so with this experiment, we did it in 2 rounds. So round 1, what we're going to call round 1, started in February. So it's as if on February 1st, it suddenly was 70 degrees out. And round 2, we started in March. And so the thing that we're looking at is chill requirements. Now let me just explain that, and then it'll become obvious. So the probability of bud break, like the likelihood that that green bud is going to pop open, for many plants, especially shrubs, requires a certain amount of chill. So it's not like they're going to be super cool listening to rock and roll. They need a certain amount of cold temperature. So if you don't have enough cold temperature, these might not leaf out. And this shape of this curve is different for every species. And so the first thing we look at is those starting in February, we can calculate for this year, or for 2017 rather, how many chill hours there were. So it's like the amount of time below a certain temperature. So we just look at the temperature record and figure out there's 1,744 chill hours. So when we look at March, those that started in March, there was 22, blah, blah, blah. Okay, so basically, the difference between round 1 and round 2 is that the round 2 plants had 500 more hours of chill. So they experienced 500 more chill hours. And that's important because there's a big difference in our results. So we'll kind of remember that that's the difference between those two. So for twig leaf out, this figure is oriented to, on the left-hand side, we have the number of days to leaf out. So basically, the shorter the thing, like the lower the box, then the earlier it happens. And along the bottom, the x-axis, are the species. And really what I want you just to know is that most of this trajectory, like this is blueberry, it goes mostly in the order we expect, except, where is it? For these 2 guys. So these 2 boxes are hazelnut, which is CA, and cherry. So in the normal, I just put this calendar up here to remind us what normal is. In the normal, black cherry is the second thing to leaf out. And hazelnut is the third thing to leaf out, like really early in the spring, early May. But in our twig experiment, where we brought them inside very early, it took them 35 days to leaf out. So that's really interesting. They were the last to leaf out. Okay, that's an interesting result, but it's the first time we ever did it. Maybe we did something wrong. But we did it with 10 twigs, and all 10 twigs were the same. Okay, so then round 2. Round 2 started in March. And the most interesting thing I want you to take home from this is our cherry. So cherry went back to being number 2. Close to huckleberry, but basically went back to being number 2. And so the only difference between round 1 and round 2 was that there were 500 more hours of chill. So this is showing us that what some research has shown from cherry data generally, that cherries are really rely on those chill requirements. And so using these data, we look at each of our study species and look at and kind of assign them a mild or moderate chill requirement. And basically what we're seeing is that cherry really requires a certain amount of chill. And so it needs more time. So, okay, that's interesting, but why is that important? Why does it matter? The thing that it shows for us is that if there is an early spring over and over again, that the cherry is not going to be able to respond. It's not going to be able to leaf out as early as it could because it hasn't had enough cold temperatures. And I'll show you one example of where we've kind of seen that in the field. So this is an example of one, or actually 2 particular years. And so in the scientific realm, this isn't enough to say it's a trend, but it's pointing to something to us. So this, once again, is 2015. So this is what we would have called a kind of normal year. This fits within that on average year of temperatures. It was an average spring. And for this figure, we have our different sites. So remember those green dots I showed you all across our map? These are the different sites. And along the bottom is the day of year. So the shorter the bars, the earlier something happens. And so day 125 is like May 3rd, I think, around there. Okay, so in a normal 2015 year, an average year, all across all the sites, the cherries leafed out right around the same time. So we're talking about breaking bud for black cherry. And so normal year, they all leaf out at the same time, great. Okay, this line, the yellow line, represents the caterpillar hatch that year. And so the eastern tent caterpillars are very susceptible to temperature, and not island-wide necessarily, but site-wide for the property at Linda Loring, they tend to hatch out on the very same day, no matter what plant they're on. And so this was, that's what it was, the reason I knew May 3rd was 125 is because that was the day they hatched. And so you can see that this is perfectly timed with when the black cherry leaves are fresh. And so those cute little baby caterpillars when they're ready to start eating, the cherry leaves will be fresh and ready and not too tough, but available to them. So that's kind of what we expect. That's how they evolved to work together. Okay, so the darker gray bars in this are 2016 data. So you can see there's a little more variation. So some cherries leafed out a little bit later, some cherries leafed out a little bit earlier. But with those error bars, and with some statistics we saw, there's no significant difference. So they're basically the same from year to year. However, when we look at the caterpillars, get ready for it. This is the take home. This is the big picture. The caterpillars in that red line hatched significantly earlier. So the caterpillars hatched 10 days earlier than the year before. There was an earlier warm temperatures that year that the caterpillars responded to. Okay, so if you're a caterpillar and you're hatching on day 115, and the trees that you are hatching on are still buds and have not leafed out, there's no food for these caterpillars. So most of you, or maybe some of you in the room might be, well, that's great because I hate those eastern tent caterpillars, and I'm glad they're starving. But what we're really interested in is this 10-day difference and what's happened to these caterpillars in that time period. And so to show you some pictures of what this looks like, when the caterpillars first hatch, they're these really, really tiny little black caterpillars. They're not ready to eat yet. They hatch, they kind of hang out for a little bit, they get warm. They're looking for these leaves, like these fresh cherry leaves, and what they were finding were these buds. Now when the caterpillars hatch and they're looking for food and they find that green tissue, they eat the buds to the point where they eat the base of the bud, and that actually prevents the tree from leafing out again. There's some evidence that shows that caterpillars eating into the bud like that is more damaging to a cherry tree than just typical defoliation that happens at that time of year. Normally, a cherry tree that gets defoliated early in the spring, by the time the caterpillars go to pupate and become moths, then the tree can leaf out again. But if the buds are all eaten out, they can't. So what we're worried about, I mean, this is one set of years of looking at this anomaly, but it's a sign to us of what could happen, and then indicates a potential ecological mismatch. So what this is, with science, when we find things like this, is when we ask more questions and design experiments about trying to get at this answer. Because if the caterpillars continue to kind of hammer on these cherries, and especially the cherry tree buds, then they could cause significant damage to one of our most common native trees on the island. And so what's next? So, we're continuing to build our baseline. As I said, I keep using the word normal, but what we're really looking at is what's our average, what's common at on the island and at Linda Loring Nature Foundation. And so we're going to continue to do field observations. The Twig experiment I illustrated, we're going to continue to do that for multiple years. This year we're going to start in January, so we'll have 3 rounds to look at earlier temperatures and the differences with that chill requirement. We'd also like to do a frost experiment, so any of the twigs that we do have that leaf out, if we then force a frost on them, how do they respond? Because different species might be able to tolerate a frost, and some species might not be able to tolerate a frost. And that will indicate to us which species that are common and native on Nantucket might be able to survive in these earlier springs. And then how will continued earlier springs affect spring leaf out and the caterpillar hatching. So if we have continued earlier and earlier springs, as we have been, the cherries haven't been able to track that earlier spring. And if the caterpillars continue to, we're interested in looking at how that affects the cherry trees, and if there's also a potential for host switching in which the caterpillars might be laying eggs on other species. And so we're looking into that as well. And so I'd just like to end with other ways that people can be involved. We'd love it if you want to be involved in our research and volunteer and collect data. But even if you didn't want to work on any of these particular projects, if you're interested in plant observation in general, I encourage you to go to the Nature's Notebook website, because they have a lot of great things. You can pick trees in your own yard, like if you have a red maple or a lilac bush or something in your own yard that you'd like to observe, and enter your own data. It really helps science nationally when the more of us collect data and then allow it to be used by others. I had to take a walk at the Linda Loring Nature Foundation. We have a Phenology trail, so there's areas where we're putting up signs with some of our research to highlight some of our research, so you can kind of check it out. You can always email me with questions. And then very finally, I want to highlight, as I mentioned earlier in one of the kind of introductory slides, Richard Premack, who did some of the work with Henry David Thoreau's journals, he's going to be here tomorrow at the Athenaeum at 7 p.m. as part of our Nantucket Biodiversity Initiative Research Conference. So he will be our keynote speaker, and he's going to be talking about Walden warming, which was the book he wrote about this research, and climate change coming to Thoreau's woods. And then we also have the Nantucket Biodiversity Initiative Research Conference going on all weekend, so I encourage anyone who's interested to register for that. And with that, I would be happy to take any questions.
Yeah.
Sorry.
If you have a question, I can come over to you with the microphone and you can ask away. Here, I'll give you the mic just so it can be on the recording.
How are you inducing chill on those plants that you were bringing in?
Right, so that was a good question. We're not actually inducing chill. So the chill that we've calculated for the plants we brought in was the chill that they naturally experienced in the field, in the natural environment before we collected them. So we know we collected all the twigs, we cut them on the same day, so we know February 1st was when we collected all the twigs. So we can calculate, based on the temperature record for this year, how many hours of chill. So it's basically below a certain temperature, how many. So sometimes people in literature calculate it by chill days, but we just did it for hours, but it's the same type of thing. And so the difference was only like what was naturally happening in March versus February. So we're still, even though it's a controlled experiment, once we're inside, the amount of chill is really dependent on just what type of winter we had. So I'll be interested to do it again this year because I've heard predictions that we're going to have a mild winter. So I think it will dramatically reduce the chill amounts for the different species. So
We've got a question here.
You left the birds out of the picture. How are the babies getting raised when cycles are off?
I'm glad you asked about birds. So because I'm not an ornithologist, I don't generally study birds, but we're interested in that cascade effect for what is eating the tent caterpillars. And actually, I think I put a little slide in because one comment I've had from birders generally is that nothing eats the eastern tent caterpillar, or that only cuckoos eat eastern tent caterpillars is another thing I've heard. So we know that there's a number of different species that eat eastern tent caterpillars when they're in a boom year. So it's like all of a sudden there's a caterpillar buffet. So it might not be their favorite food, but if it's the most abundant food, they'll eat them for sure. So these are just a few pictures. We have cuckoos. The one up front that says kind flow, that was, if you guys know Spruce, he took that photo of a cedar waxwing eating an eastern tent caterpillar. Orioles, chickadees, and the one in the low middle is a pine warbler. Okay, Harvey's nodding, so I'm going to just say yeah. Okay. But so we haven't tied it directly yet to any particular individual species, but there are a number of bird species who take advantage of the caterpillars.
Come over to you.
Thank you. Hi, Sarah.
Hi, Dana.
I'm curious about what kind of research you've been doing to see if there's any documentary evidence in the NHA archives of any kind of temperature data or anything like that that might serve as a baseline for an early period of time.
I'm so glad you brought that up, Janet, because one thing about presenting here was I'm very interested in historical records, and I know I'm excited to talk about with Richard Premack when he's here to talk about some of their gleaning of the historical records. He didn't find Thoreau's notebook and then suddenly decide to do this research. Like I know from Richard, he wanted to do this research and was trying to find the best historical record, and actually birds was the first thing that came up because of the history of the Christmas bird count data and that kind of thing. It would be really interesting to kind of go through the NHA archives to see where we have either temperature or species records, because that's where we can really see the difference. I mean, I think we can more easily look at temperature records over time to look at how Nantucket climate has changed, but how species have adapted or not adapted to that change and changed their either migration or leaf out or thing would be really interesting. A lot of other areas look at herbarium records, so you can look at the flowering. If you have an herbarium specimen of a flower and it's like a certain date, you know that it at least flowered at that time. But with a lot of that kind of record, you need a lot of records like that. So that's why people do kind of bigger scale things. We have a great herbarium at Mariah Mitchell Association on the island, and the UMass Boston Field Station has a great additional herbarium. But I don't think there's the number of specimens that we would need. We've talked about it locally with the Nantucket Biodiversity Initiative. Some people have talked about Edith Andrews' records because if you knew Edith, she was obviously an amazing ornithologist, but she kept records on a lot of other things as well. So I think those are the kinds of historical records we're interested in. I know, if anyone knows Aldo Leopold and the Sand County Almanac he's famous for, he also kept extensive records of his property. And so his daughter and then his granddaughter have continued these records. So there's actually a really long historical record there too. And so I think if we can kind of piece together some of these journals and kind of pull the data from the historic writings, I think they're kind of a really interesting marriage of history and science, I think. So thank you for that question. It's kind of spot on of what we're interested in too.
Got one last question over here, and then we'll wrap things up.
Hi, thanks. I'm actually hailing from Oregon, a native of Massachusetts, but I've lived out in the Pacific Northwest for over 30 years now, and I live in the Columbia Gorge area, and there's about, I would say, maybe over 12,000 acres of cherry trees in the area where I live, and personally I'd grown for 20 years cherries. So I think it's interesting to hear what you're doing here, and I'd be fascinated to learn. I know Oregon State is a land grant university, but because it's a production that is mostly involved in the fruit and making money, there's huge records of production of the fruit trees in the valley where I live, and there's a sister city in Japan where there's also fruit, which I'm sure that those records are from, but I'd be interested to know if anyone had ever looked at some of the historical data from there. I mean, I'll do it myself when I get back, but it's just interesting to see the mapping that you showed where I noticed in the Pacific Northwest the spring wasn't shown as earlier, though that's not my experience.
Yeah, I think so. So just caveat, I was at Oregon before I came here. So I lived in Corvallis, where OSU is, and the reason cherries are studied so extensively, whether they're ornamental varieties or fruit production varieties or native varieties, is that for all those reasons I just said. Ornamentally, they're very interesting. They're culturally significant. So there actually are a lot of records, phenological records for cherries, and the National Phenology Network is a national program, and they highlight certain species they want people to study. And so cherries are number one on there because there is a historic record for them. So if you're interested, I would look at the National Phenology Network, and anyone can just kind of peruse through the data and look at different things you want to look at, but you can see in your area too what's being done. I think because this was 2017, that early spring map I showed, I think it was because, remember, there were significant rains last year. I mean, we're all thinking of the fires this year, like now, but in last winter, there was so much rain in California and Southern Oregon. I remember all like the desert wildflowers were blooming. That's like in the news a lot. So that was, I think that made for a late spring because it was so wet. I'm not 100% sure, but that was just kind of my thought of why that particular year was late. And so that's why looking at any one year, it's not a trend, but it's, I like to put that on there just to see how there's such variation in our country and even in the regions. And when we say, spring's happening earlier and earlier, well, year to year, there's going to be some variation in what we're actually seeing. Yeah. So I'd be interested to hear more because I think a lot of phenology research, because people are working with whatever historical data is available, is coming from either agriculture or I know there's a lot being done in wineries because there's vineyard records of grapes ripening. So, we talk about spring leaf out because people are interested in green up, but flowers are the primary phenophase for cherries, and then for things like grapes and the winery industry, it's ripening. So it just is whatever people have observed, basically. So.
Well, let's have another big round of applause. A big thank you.
Thank you so much. I really appreciate it.
We will be back here next week with Dr. Charles Stormy Mayo from the Center of Coastal Studies. So we hope to see you back here next week. He'll be giving a talk on right whales. Thank you all.